Good morning, everyone. Thank you, Ethan. I want to actually first thank the amazing staff at the SID and the fantastic team, the Scientific Planning Committee, that has done a great job so far, and the meeting looks fantastic. Also want to thank them for the opportunity, for giving me this opportunity to talk. I hope by the end of this 30 minutes that you will be convinced that mouse models are useful in studying psoriasis, pathogenesis, as well as potentially in other diseases. All right, that one's not working. So these are my disclosures, and this is what I'm talking about. So psoriasis pathogenesis is rather complicated, and this is a cartoon made by a postdoc in our department, David Solar, and I hope you can appreciate that there's pretty much almost every single cell type and every single cytokine that is known to man or woman on this cartoon. And trying to understand how the disease actually happens, how we can drive it into remission, has been a long process. And I think if you look at the former Eugene Farber lecture givers, these gentlemen, these scientists, I hope you can appreciate the legacy behind this lecture, because these gentlemen really contributed to a lot of our knowledge that led into that cartoon that I just showed you, whether it was the genetics of the disease, the comorbidities that may be related to psoriasis, the immunology, the ability to do xenograft approaches, the amazing translation that we now know targeting specific cell types and cytokines and how they contribute to disease pathogenesis. And Chris Griffiths, who last year talked about how he's created this group in Manchester called Impact Psoriasis that really takes into account the entire patient. So how do we model this? How do we learn from models? We can knock out specific cytokines and study their effects. We can knock out specific cell types, T cells, dendritic cells, look at what happens. We know that there's mice that overexpress either specific transcripts or signaling pathway molecules. We can use xenograft approaches, take patient skin, put it onto a mouse, study what happens. There are cell-specific approaches now where we can actually do conditionals. We can turn a gene on and off in a cell-specific manner. There are acute models, the application of Aldara, the intradermal injection of cytokines. And then there's combination approaches where we can take bone marrow from knockout mice, put it into another mouse and study the circulating cell contributions versus the skin contributions. As we've learned more about mouse or more about psoriasis pathogenesis, the number of mouse models has continued to increase. In fact, right now, there's more than 45 unique mouse models modeling psoriasis out there. One of them, the Aldara topical application model that Errol Prenz and Eric Lubertz published in 2009, I hope you can appreciate, has escalated the publications in psoriasis mouse model work, more than 130. And in fact, 74 unique either knockout or transgenic mice have had Aldara put onto their skin and then the phenotype studied. A little bit of perspective. When we take all of this mouse model information together and we plug it into Cytoscape and do Klugo analysis on it, I hope you can appreciate that everything really holds hands. Everything has some level of synergy, whether it is the activation of immune cells, the regulation of transcription factors, or protein signaling cascades, or ultimately the positive regulation of epithelial cells, which makes sense. These all phenotypically look the same. Now, long before there was any type of ingenuity pathways analysis, biosystems approaches, or Cytoscape, or even doing multi-omics approaches and creating large, big data sets, Gene Farber was around 36 years as the department chair at Stanford in dermatology. And he was an idea man. This guy had lots of ideas, and he really sort of led the charge of taking clinical observations in the clinic and bringing them back to the lab and trying to study them. I never met him. I was introduced to him at this meeting, the Montana meeting in 2007. And Brian Nikoloff was standing up here like I am right now, and he was telling stories about working with Gene Farber when they were at Stanford. And I was mesmerized. I was sitting in the back corner, and Brian was talking about how Gene, a patient would come into the clinic, and they would have their plaque completely resolved on the side of their leg that they had undergone some sort of surgical denervation, knee surgery. And I was like, wow, my PhD is in neurobiology. No one understood what the phenomena, how it happened. So I went home being the good, I still think of myself as a student, but as a good young PI, and I started looking at Gene's early work, looking at the role and these ideas about stress and symmetry and psoriasis and the role of the nervous system. Look at these years. 86, 89, 90. These ideas were really expounded very early, and just a little perspective. I just graduated from high school. And yeah, you can see I come by my crazy naturally. That is my family. So looking at these papers, you can see what Gene was proposing, that perhaps the symmetry of psoriasis was caused because of sensory nerve innervation. And that in these patients that had knee surgery and their plaque went away, perhaps it was because of nerve contributions. And he proposed this sort of cascade of events with substance P being at the top of them. Now, at the same time, at this very same Montagna meeting, I was presenting the very first mouse model that I ever engineered, the KC-TY2 mouse. And I was being very, following Brian's suggestions, and I was going through it, and I was like, okay, yeah, has the same physical characteristics. It's got angiogenesis and epidermal differentiation characteristics of psoriasis. It's a Th1, Th17 model, and it's responsive to immunosuppression. At the same time, I had been recruited into dermatology by Kevin Cooper, trial by fire. I wrote a grant with them, and this is the grant, old school, right? Paper, the court. Notice Cooper, Dr. Cooper looks fantastic. The rest of us are like, no sleep that night. This grant got funded, and it just gave me an amazing opportunity to study psoriasis pathogenesis. Essentially, all models are wrong, but some are useful. Could we use our mouse models to study what Dr. Farber was talking about, what Brian talked about with such great passion at these meetings? So I apologize for my art. I'm not talented in this realm, but that's okay. This is what we did. We took a mouse. We made a midline incision down its back. We basically opened up the flaps of the dorsal skin. You can visualize the cutaneous nerves. On one side, we exotomized them. In fact, you can take them right out. The other side, we left untouched. We closed the mouse up with staples. You immediately can tell if the denervation was effective, because if you poke the mouse with a pin, it won't turn towards that stimulus. We waited for 10 days. We confirmed the region of denervation, and we harvested skin from both sides. Another story. I presented this at some research in progress meeting in our department. And dermatologists are very visual people, and this comment totally exemplifies that. So I showed this slide, and Dr. Cooper said, wow, your denervation changed the vibrissae of your mouse model. Our denervation worked. When we looked at PGP 9.5, a global nerve cell marker in the denervated skin, the nerves were gone. And remarkably, I hope you can appreciate, this is the sham-operated side, this is the denervated operated side of the same mouse, and there was a significant reduction in the skin thickness. And that's quantified here across different mouse models or different specific animals. And it's not unique just to this one model. We showed similar outcomes in both the imiquimod topical model as well as the IL-21 intradermal cytokine model. The skin thickness was thinner in denervated skin, either when you put it onto denervated skin or you denervated the skin after the skin inflammation was already established. The dendritic cells disappeared very quickly, back to control mouse levels. The CD4 positive T cells also decreased. And when we looked at this over a time period, the dendritic cells actually changed first, very rapidly, followed by decreases in cutaneous IL-23 expression, then changes in the epidermal thickness, and then finally the decrease in the T cells. Now, back to medical school or graduate school. The cell bodies of the skin, nerves, live back in the dorsal root ganglia that lies along your spine. Kind of hard to study them in people. Can't harvest them, can't look at transcript analysis, and it's hard to get them from cadavers. Great thing about mice is you can go in, harvest all that tissue, and look at transcript changes. And what we discovered was that molecule that Dr. Farber talked about, substance P, was elevated, as was another one, CGRP, calcitonin gene-related peptide. Two candidates that now we could use to try and mechanistically figure out why eliminating the nerves got rid of the disease. So this is what we did. We took denervated skin, mice we did the denervation on, and we intradermally put the peptides back in. Conversely, we kept mice with their nerves intact and inhibited those specific peptides. And then we looked at the same outcomes that I just told you about. So this slide is probably a summary of 100 mice, and what I want you to appreciate from it is that the neuropeptides have a cell-specific effect. CGRP has no effect on dendritic cells, but can influence both the epidermal thickness and T cell numbers. Substance P, on the other hand, has no effect on the epithelium, but can influence the dendritic cells and the T cells. And so we're working now to try and figure out whether these effects are specific, direct effects, or indirect effects. There is a poster by Adriana Laragina and Tina Sumpner looking at T cells and substance P neurokinin-1 receptor interactions at this meeting. If this is interesting to you, please go see it. They do great work. All right, so the cool thing about publishing a paper like this and being a PhD is patients are very savvy and smart these days. And they see these, they find this somehow. This is an email I got from a patient, and she sent it to me along with a picture. So this is kind of cool for someone, a basic scientist like me. This is the knee. She had knee replacement surgery. She used to have plaque on both sides, and all of her plaque resolved on the knee that she had operated on. Super cool. All right, so now this approach and our findings have now been broadened beyond psoriasis. This is not just unique to psoriasiform skin inflammation. Sunny Wong published a really cool paper showing that basal cell carcinoma development is also dependent on nerve-derived factors. Similarly, Dan Kaplan and Sakine also showed that sensory nerve-derived peptides are critical for dermal dendritic cell IL-20 and fighting off Candida. And there's more posters at this meeting, including one from Ethan, looking at the role of the nervous system in inflammation and atopic mast cell survival, as well as looking at norepinephrine and its ability to drive T cell activation by Rick Granstein's group. All right, so Steve, who was a med student at the time he was working on this, is now a PI and a dermatologist at Mass General, presented this work at yet another Montagna meeting. The Montagna meetings hold great meaning to me. They're a wonderful opportunity to do good science, present good science, and interact with brilliant people in a small environment. And he presented his work. And Dr. Gilchrist stood up at the question section, part of his presentation, and said, so, you know, neurotoxins have this ability to inhibit nerve release of substance P and CGRP. Have you thought about trying a chemical denervation? And so we did that. We went back to the lab. We took our mice. We injected them with a single injection of neurotoxin. And then we waited either 2 or 6 weeks later. And we found the same effect. Skin disease improved in these mice that were chemically denervated. And then in collaboration with Aaron Gilbert, we were able to translate this into a patient and show that a recalcitrant plaque on this patient's buttock, injected with, again, a single intradermal injection of toxin, resolved the plaque within 3 weeks, sustained it clear out to 7 months, and then reappeared in the same location. And Maria Hordinsky has a poster showing another clinical study showing efficacy of neurotoxin at this meeting. So just a plug, this year's Montana meeting is looking at the role of nerves, itch, pain, touch, and pleasure. If this is something you're interested in, please come. Abstract deadline is coming up. If you're a trainee, there are Farber travel awards for you, so think about coming to the meeting. All right, so Facebook is reminding me that I clearly use the word psoriasis in my social media presence frequently, since it's the largest word. And then I also apparently use the term best team ever. I love my job. Find a cure. And just so you know, I'm not completely focused on work all the time. I do mention Theo, my dog, and Katie, my 16-year-old neighbor who comes over to my house and hangs out. All right, so while we're doing all this nerve work, Joel Gelfand and Nehal Mehta are talking about this cardiovascular disease risk in patients, that they're dying 5 to 7 years younger, that it's probably because of MI, heart attack, stroke, DVT. And I came back to this thinking, you know, essentially all models are wrong, but some are useful. Could we use our mouse model and show a cause-effect relationship between the two conditions? And, in fact, we were able to do that. So in animals with skin inflammation, if you age them out to beyond a year, about a third of them in this model develop vascular inflammation in the aortas. They also become very prothrombotic. So as early as 10 weeks of age, they clot very quickly in an experimental thrombosis model. And prior to all of this occurring, they have elevated monocytosis, the CD11b Ly6C high population of cells. This suggested to us that the skin inflammation could cause the cardiovascular comorbidities. And so we turned the gene off. The way we engineered the mice is we can turn the gene off and get rid of the skin disease. And it also got rid of the cardiovascular inflammation in the aorta and brought the clotting times back to control mouse levels. Lots of common molecules involved in both diseases. This one jumped out at us, S100A8A9, calprotectin. Why? In cardiovascular literature, it correlates and corresponds to increasing risk, HDL, CRP. If you knock it out of a mouse model, that mouse has a lengthened time to clot. It's protected. We know it's up in psoriasis skin. It's up in the epidermis. It's up in lesional skin. We also knew it was up in the KC-TY2 mouse, both in the skin and the serum. But what about monocytes? You know, they're up in the mouse, but what about patients? In the cardiac patients, it's actually a better independent predictor of a stroke or a cardiac event or death than CRP. And work recently done by Jackie Golden in Kevin Cooper's lab showed that these same inflammatory monocytes are up in psoriasis patients, this intermediate population, CD14 high, CD16 mid, and it correlates with skin severity. So we hypothesized that maybe S100, A8A9, MRP14 would be the miracle transcript. We knock it out, it should improve everything. So to the young people out here, this is 10 generations of back crossing the KC-TY2 mice to the Black 6 background before we could back cross them to the S100 A9 MRP14 knockout mice. And I hope you can appreciate nothing changed. 3 years of work, God knows how much money in animal housing. Nothing changed. The skin's still inflamed. They look the same. The skin's still thick. And worse, the clotting time didn't change. Why? Something in the skin must still be driving it. And we looked at the skin, at the pro-inflammatory transcripts, and we showed this trend of continuing to increase cytokines for IL-23, IL-17, and IL-6 at the transcript and at the protein level. So we took the KC-TY2 MRP14 knockout mice and we blocked, using P19 antibodies, both of these. Skin disease gone, which is good. That's consistent with what we see in psoriasis patients. Remarkably, the thrombosis clotting times increased back to control mouse levels, suggesting that treatment of the skin disease may be protective of the cardiovascular event. Now, what was interesting was that IL-6 in the skin came down. And remember, IL-6 was one of those transcripts that was also staying up in the transgenic knockouts. The murine antibodies to inhibit this cytokine are awful. So we went back and we back-crossed these mice to the IL-6 knockout animals. And yet again, no improvement in the skin. But this was okay. We didn't expect to see it because we know psoriasis patients treated with the IL-6 inhibitors, their skin doesn't get better. Their joints do, but their skin doesn't. But check out what happened to the thrombosis. It also improved. When we looked at the monocytosis and we tried to sort of correlate it to short clotting, which is bad, long clotting, which is good, what we found was in the mice that had short clotting, the monocytes were up compared to the control mice. In the animals where their clotting was lengthened, the monocytes decreased. So Jackie, who was doing this, got excited, and Tom McCormick, our collaborator, was excited, and I looked at this and I was like, this is not exciting. The delta's not big. I mean, sure, it might be meaningful, but it doesn't look very exciting to me. What about other cells in the circulation? So when we looked at the CBCs of these animals, what we found was the platelets were up and the neutrophils were up in the mice that clotted quickly. And in the IL-6 knockout mice where we got complete protection, even though the skin was still inflamed, the neutrophils and the platelets came back down to control most levels. I have other data showing efficacy with biologics and looking at these same outcome parameters. Come see the poster, come hear the talk. I don't want to steal the thunder, so just a plug for that. Now remember, we have other mouse models in our lab. Is this unique to the KC-TY2 mouse? Would we see it with the Aldara mouse? So we had this new mouse in our lab, a chronic spontaneous model overexpressing IL-17 in the C in the skin. It gets great skin inflammation, and we compared it to the Aldara model, 5 days of skin inflammation. The only mouse that clotted quickly was the mouse that had chronic skin inflammation for 10 weeks. The Aldara mouse didn't clot any differently. I think this is good. It means if you have a skin infection or an acute inflammatory skin reaction, you're not going to keel over and die of a cardiac event. Now, when we looked at the monocytes, what we determined was the monocytes really seemed to correspond better to the level of skin inflammation. The neutrophils, however, were only elevated in the mouse that was prothrombotic. Nehal Mehta just published a paper in ATVB showing that circulating neutrophils are up in psoriasis patients and that neutrophil-specific proteins correlate really well with vascular inflammation. So it's possible there's a role for neutrophils in driving the comorbidities of this disease. IL-6 rescue, not just unique to the KC-TY2 mouse. When we back-cross the 17C mouse to the IL-6 knockout, skin disease still there, thrombosis clotting times come back to normal. So we're being able to now separate out skin inflammation, things that are coming out of the skin that can influence distant vessels, and separate out the two. So what does this mean for psoriasis patients? So we know 6 inhibitor doesn't work for skin. In fact, Alice Gottlieb and Philip Mees have this paper that's available as an EPUB showing with clazakizumab the same thing. But what's interesting across all of these papers is, guess what goes down in the CBCs of these patients? Neutrophils, platelets, and serum CRP. So there's a lot more to do going forward to look at the role of IL-6 and what it may contribute to pathogenesis at the cardiovascular level of psoriasis patients. So back to the modeling mayhem question. Is there utility? I think so, yes. I think you can use mouse models to get at mechanism of action of things that we didn't understand. I think we can use them to model comorbidities. I talked about cardiovascular disease, but you could look at kidney, liver, depression. I think you can develop new mouse models around key signatures that you identify in patient skin or blood, model them and study them in an animal model. You can do all of these cool experiments where you can introduce bone marrow from a knockout into the mice and look at how cells actually communicate. And you can take your mouse model, learn something from it, validate it in patients, and then go back and generate a new mouse model. So in the last 3 3/4 minutes, I want to speak to both the senior people in this room and the junior people. it takes a village to raise a scientist. Mentors, role models, champions. This is your home society. For the junior people, we want you to be successful. We are here to enable your success. Come talk to us. I did. I talked to these people, and I want to thank them. Paul Bergstresser, Amy Paller, Rich Gallo, Yoni Wito, John McGrath, Jim Kruger, Steve Katz, John Voorhees, Brian Nikoloff, Barbara Gilchrist, Chris Griffiths, Angela Cristiano, JT Elder, Kevin Cooper, my department chair, the man who had the courage and the vision to pull me out of an anatomy department into dermatology. Tom McCormick, my longtime collaborator, my immunology expert, the person who never makes me think I have a crazy idea or that I have a stupid question. It takes a village. And clearly, I didn't do all of this work. I have to thank my team. Best team ever. I am a huge believer of that. Doina and E have been with me for almost a decade. They are the anchors of my team. They are the ones who get everyone into the lab and being productive quickly. The work that I talked about today, for a large part, was done by Steve Ostrowski when he was a medical student. The cardiovascular stuff was a huge team effort across Marina Lee, Jackie Golden, Sean Dawes, as well as these two remarkable women, my collaborators in the Division of Cardiology. And you can see we do have fun. We don't just work. We are appreciative of good food and alcohol. So I want to thank you all for your attention. And I hope you all get engaged in this wonderful society. Thank you.
So Dr. Ward, that was a fabulous presentation. Covered a huge amount of ground, demonstrates the complexity and for your troubles. And she's already told me she may be using this at the mall.
New handbag.
Dr. Ward, thank you.
Thank you.